All right, thanks. So I'm Daniel Jensen. I'm at JPL, and I'm here to show you some of the data sets that we have published or in progress that we derive from AVIRIS-NG, the Airborne Visible Infrared Imaging Spectrometer Next Generation, for our vegetation products. So just a little background on what imaging spectroscopy or hyperspectral remote sensing is, as people usually refer to it. These are continuous radiance measurements that we can usually process to surface reflectance at each pixel that the airborne instrument gathers. So the full data set that is compiled can be thought of as this image cube that you can see in the lower right that estimates visible to shortwave infrared reflectance properties at each pixel. So like, for example, at this image, if you look at a vegetation pixel, you can pluck out the data and plot the reflectance values across all 425 bands that we have here. And this is what a typical vegetation spectrum looks like. And this is old data, so there's some other noisy artifacts still lingering in there. The newer data is generally a little bit better. But these spectral characteristics that you can derive from these data, they can be closely associated with plant structural and biochemical properties. So things like canopy water content, photosynthetic capability, and other like canopy nutrient traits, things like that. So for AVIRIS-NG, which flew on the B200 King Air that you can see pictured here, it measures radiance, again, which we process to reflectance for these purposes, from 380 to 2500 nanometers at 5 nanometer sampling. So that spans 425 bands, and our data here, it captured at a 5-meter spatial resolution. So this is sort of the processing, the whole range of processes that we go through to generate our L1, L2, L2B, and L3 products. So we start with the radiance products that you can see on the left. This is what the sensor sees in microwatts per nanometer per steradian per centimeter squared. And that's what's processed into surface reflectance with the atmospheric correction algorithm. So that's the second panel there. Again, a typical vegetation reflectance spectrum. We added an additional processing step to correct bidirectional reflectance distribution function effects over the land so that we get consistent spectra, flight line to flight line as they're overlapping, as well as sun glint over the water pixels. So that's a L2B dataset. And then that is the dataset that we use to generate our L3 products, such as water quality maps and our vegetation maps. And this is just what some of that looks like. So the top right here is the full range of coverage that was flown in spring 2021. So in that deployment, we collected all of those lines along with additional water quality lines that I think you saw before that crisscross these vegetation boxes. And at the bottom is some BRDF and glint corrected mosaics, the L2B data sets from that spring campaign. So those are, again, what we use to generate our L3 products, the vegetation type maps and above ground biomass maps, which I'll show you here today. So we collected those two mosaics again in August, and also following Hurricane Ida, we collected the whole range of all those boxes. So for vegetation mapping, this is our general process. We start with the in situ points from two sources primarily. The vegetation survey conducted by our colleagues at FIU, as well as the Louisiana Coastwide Reference Monitoring System vegetation plot data. So everywhere where the plot had a dominant species, that was coincident with our AVIRIS-NG coverage, we used the coincident pixel spectrum along with our Delta X survey. So that constituted 504 points, and that was the basis that we used to construct our spectral library for this mosaic. So we conglomerated all those vegetation data into these different classes. So forest, broadleaf herbaceous, which are freshwater dominant, Salt marsh grasses, and some of these species also include brackish species, freshwater grasses, these tall grasses like Phragmites and Typha that are structurally a lot different than the former two, and then aquatic vegetation and soil. So our classification algorithm here, the first step was to do a principal component analysis trained on that spectral library, not the individual mosaics, because if we did that, then our results would be different between our different data sets. We applied that PCA rotation and selected the PCs for classification input. So this was a key step because some of these principal components, when you apply it to imagery, they'll pick up on things that can vary between flight lines, like different atmospheric effects. This is one of the challenges of remote sensing, especially in very humid environments like August in Louisiana, because the atmospheric conditions can be very variable and change day to day and also hour to hour. So this is an example. The top is a good principal component that is very consistent across flight lines and kind of helps eliminate any of these cross-track discrepancies. But the bottom one, while it is a significant component, you see like the bottom collection, these lines were collected, I believe, the day before compared to the top ones, and you see that a big line cutting through it, and that would translate into errors in your classification. So from our PCA analysis, we selected 13 final components that were consistent across the whole domain, and together they explain 99.86% of the variance in our 300 utilized bands. So those are the data that we fed into a random forest classification model. And we did that by training the model on a randomized selection of 2/3 of all of our vegetation and spectra pairs within each class. And these are the results from that model. So the map here is the final complete vegetation type map. You can see the Atchafalaya is very dominated by broadleaf herbaceous and forest classes. And then in the Terrebonne, you see this gradient from broadleaf herbaceous down into salt marsh grass to the southeast. You can also see some forest class where there's mangroves sort of expanding down to the southern tip there. And then I put this inset here in the top right to show just some of a little more detail and also some of the patchwork involved here. August in Louisiana is obviously there's no cloud-free days, so there's some clouds and shadows present. Where we had clouds and shadows in our August data, we filled it in generally with the September data after Hurricane Ida hit. And that did a pretty good job of consistently classifying those types and filling in all our gaps. So we have total coverage at the peak vegetation season. And this is our validation for this these vegetation products. Again, this is based on the 1/3 randomized subset, 167 of the 504 points. And this gives us an overall accuracy of 0.85 and a Cohen's kappa value of 0.81. So that's pretty good for compared to most published vegetation classifications. So moving on from the vegetation type map into our above ground biomass products, we use these same spectra coincident with the vegetation survey that the FIU folks did to model herbaceous above ground biomass as a function of those reflecting spectra. That's across the spring samples as well as the August samples. So there's 42 vegetation spectra pairs in each of those collections. Not to get too into the weeds on the algorithm here, but again, because the August data was so humid, there was some noise artifacts in the near infrared ranges here that's highlighted in red in the figure. So we developed a different algorithm that sort of imputes these values because they're very important spectral regions that are indicative of how much water is in the plant canopy. And that's obviously an important facet for estimating biomass just from these spectra alone. So with those corrected, you can see the bottom what the corrected spectrum looks like, and then also one of the what one of the corrected image bands in the lower right looks like compared to its before state. So we again used a random forest regression model based on those corrected data to make our final maps. And the data from this and the whole process and analysis, this is in review at remote sensing of environment. Before I get to the final maps, just wanted to show some of our modeling, comparison, and analysis, and show how we arrived at this final random forest model. So our V1 data was done with an uncorrected partial least squares regression approach, and that's the call out in sets A. You can see in some of these areas there's a lot of noise, especially at the edges of the flight lines, some errant values where the overlapping areas don't match up. Some of these weird atmospheric artifacts kind of bled through. B is a partially squares regression approach using the corrected data, which is better, but we still have some of those same effects present, and also it doesn't seem to capture a lot of the variability. And then the last one, C, is from our final maps, and you can actually see the machine learning based model accounts for a lot of those cross-track discrepancies a lot better. And if you go down to like individual vegetation stand results, you can see a lot more detail. So some bigger like grass stands, for example, look like higher biomass, as they should in reality, whereas they don't in the other approaches. So these are our vegetation, herbaceous vegetation above ground biomass products from both the early biomass season and the peak season. The overall model achieves an R squared of 0.89 and a mean absolute error of 109.3 grams per meter squared. And then for validation, we used a simple leave one out cross-validation approach to look at the sort of direction and magnitude of the errors. And you can see the results here in the scatter plot. And if you want more detail, all that data is also on the ORNL DAC page for this data set. So yeah, with that, just acknowledge the project itself, and here's the links where all the relevant data sets are available. So the first one here is our above ground biomass maps. The vegetation map will be forthcoming, and then the second one here is for the L2 surface reflectance mosaics and flight lines, which also I want to note includes data masks. So it's land water masks and clouds and shadows are separated out by pixels if you want to do your own analysis. And then the last one here is the field data set that we based all this on. Yes, and that's it. So thank you.